You know, usually when we talk about fixing a health problem, there's this expectation of precision, like taking your car to a mechanic. If you have headache, you take aspirin and the headache goes away. It's a clean, direct intervention, right?
Right, yeah. We expect a straightforward action and reaction, like a really clear mechanism.
But then you step into the world of the human microbiome and, well, all that precision just completely evaporates. You go to the store, you buy a phenomenally expensive probiotic pill that promises to fix your gut, you swallow it, and absolutely nothing happens.
Or worse, you just get incredibly bloated.
Exactly. And for the longest time, the reason why has been this massive mystery. Because until now, microbiome science has essentially been like taking a census of a massive chaotic city. You're counting all the residents, but you have absolutely no idea what jobs those people do or what they like to eat or who they are competing with for resources.
Yeah, it is the absolute definition of flying blind. I mean, we've spent years expertly describing what is sitting in the gut, but we haven't actually known what it's actively doing, let alone how to reliably change its behavior.
Well, a new paper out of Cell by Moyne et al. Has finally cracked the code on this. They built a framework called MIND, which stands for microbial interaction and niche determination. And the mission of this deep dive today is to go sentence by sentence, unpacking every conceivable detail of how scientists have finally learned to actually predict and control microscopic ecosystems, like programming a computer.
What's fascinating here is that the MIND framework moves the entire field away from mere correlation. You know, before we were just looking at what bacteria happen to be sitting next to each other. Now we're looking at strict causation. We finally have the mechanistic truth required to manipulate these ecosystems with surgical precision.
To really grasp how massive a leap this is, we have to start with how scientists actually look at the data inside a microbiome. There are basically 3 layers of information, which researchers call the omics.
Yes, and understanding these 3 omics layers is crucial to seeing why past approaches failed so hard. So first, you have metagenomics. This is essentially looking at the DNA of the entire microbial community. It represents pure genetic potential.
So it just tells you everything the microbes in that environment could possibly do if they wanted to.
Exactly, just potential. Second, you have metatranscriptomics, which looks at the messenger RNA, or mRNA. When a cell decides it wants to use a specific gene from its DNA, it transcribes it into mRNA. So this second layer tells us what messages are currently being sent out by the nucleus.
But the authors point out a major flaw in just looking at the messages.
Right.
Because just sending a work order doesn't mean the work actually gets done.
Precisely. In biological systems, messages degrade, conditions change, resources run out. And that brings us to the third, and frankly most critical, layer metatranslotomics. Specifically, they relied on a technique called metaribosec.
Okay, let's unpack this. What does that actually look like inside the cell?
Well, ribosomes are the massive cellular machines that physically grab onto the mRNA and read it to build proteins. By sequencing only the tiny fragments of mRNA that are currently locked inside an active ribosome, you aren't just seeing what messages were sent. You are seeing the actual active protein synthesis happening in real time.
The analogy that makes this click perfectly for me is imagining the microbiome as a massive, bustling food court. The metagenomics, the DNA layer, is the master menu for every single restaurant. It's what they're theoretically capable of cooking.
Right.
Then the metatranscriptomics, the mRNA, are the order tickets being sent from the waiters back to the kitchen. But kitchens get backed up, right? Orders get dropped. So metatranslotomics, the metaribosec layer, is the equivalent of walking right past the waiters, going directly to the stoves, and looking into the pans to see what the chefs are actively cooking right this very second.
That is a phenomenal way to visualize it. I love that. And that exact distinction leads us directly to the core metric that makes the entire MIND framework possible, TE, or translational efficiency. The researchers calculate TE by taking the active metaribosec data, what's in the pan, and dividing it by the metaRNA-sec data, the order tickets.
So why is that specific division, like that ratio, so vital to understanding these microbes?
Because making proteins is incredibly expensive for a microscopic cell. It consumes massive amounts of cellular energy in the form of ATP. Microbes do not have an endless supply of ATP, so they have to ruthlessly prioritize.
Oh, wow.
Yeah, they will only fully translate the mRNAs that are absolutely essential for their immediate survival in that exact moment. So if a specific gene has a high TE, it means it has a very high biological priority. It tells us exactly what the microbe actually cares about.
It completely strips away the hypothetical. It's the difference between what a microbe claims it can do on its resume and what it's actually bleeding energy to accomplish today. So to summarize this first subtopic, DNA is potential, But TE reveals the active reality. Think about your own life for a second. If someone calculated your personal translational efficiency, what tasks are getting all your energy right now?
That's a great question. And once we have that snapshot, once we know what a microbe is prioritizing via TE, we can map out the whole ecosystem using ecological guilds.
Yields, like in macroecology.
Exactly. In macroecology, a guild is a group of organisms that exploit the same class of environmental resources. Think of lions and cheetahs on the savanna. They are in the same guild because they hunt the same prey. The mind framework applies this microscopically.
So if two different microbes prioritize the exact same metabolic pathways, meaning they have highly similar TE profiles, they belong to the same functional guild.
Yes. And if they're in the same guild, they are guaranteed to compete fiercely for the same resources. We're no longer looking at who is related to who their family tree. We are strictly looking at who is trying to take the exact same plate of food.
To prove this actually works, they didn't just run computer simulations, right? They built a physical 16-member synthetic community, a syncom, assembled from bacteria found on switchgrass roots.
Right. They grew this specific community together in a complex liquid environment called 0.1x R2A medium. They established a baseline and then started running dropout experiments.
And a dropout experiment is where they literally just remove one single microbe from the mix and watch how the other 15 react.
Precisely. And using only the baseline TE profiles, their algorithm predicted who would compete with whom, achieving 87.5% sensitivity and 71% specificity.
The sheer scale of the reactions is what caught my attention. When they dropped out a microbe called Mucilaginibacter, its TE-predicted competitor, Ctenophaga, didn't just do a little better. It exploded, multiplying by 6.8 times its original population.
Yeah, it just took over.
And in another test, they removed Burkholderia, and suddenly a microbe called Rhizobium shot up by threefold. Remove the rival, and the competitor immediately steps in to consume all the newly available resources.
Oh, yeah.
But wait, when you say they are competing, what does that actually look like? Are they actively attacking each other, like microscopic warfare with toxins, or is it just a race to consume the resources faster?
That distinction is incredibly important. It's the difference between interference competition, where they produce toxins to actively kill each other, and exploitative competition, where they simply out-eat each other. To find out, they ran a spot-on-lawn assay.
How does that assay work exactly?
Essentially, you take a petri dish and grow a solid lawn of one specific microbe. Then you drop a concentrated spot of a second microbe right in the middle of it. If the second microbe produces a toxin, you'll see a clear halo, a zone of inhibition form around it where the first microbe was killed off.
Oh, I see.
Yeah, and they systematically tested all 256 possible pairwise combinations of those 16 microbes to look for these toxic interactions.
256 microscopic cage matches, basically.
Yes, and out of all 256 matchups, they found only one single pair that showed interference competition. Ctenophaga actively produced a substance that inhibited Mucilaginibacter. For every single other pair, the competition was purely exploitative.
Wow. No biochemical weapons. They were simply racing to consume the exact same food. Okay, let's do a quick 5-minute recap and bullet summary. Bullet 1. DNA is potential, but TE is active reality. Bullet 2. Microbes with similar TE profiles form competitive guilds. Bullet 3. Removing a microbe directly boosts its specific guild rival.
That sums it up perfectly.
So it's mostly just a high-stakes eating contest, not a battlefield. If you were a microbe, would you just eat faster, or would you be the one throwing biochemical elbows?
I'd probably just eat faster, honestly. But that brings us to the second major pillar of mind niche determination. If we know they are racing, how do we figure out exactly what they're racing for?
Right. How do we determine the menu?
They zoomed in on the TE data again, but looked specifically at the genes that encode for import proteins, which are also known as transporters.
Transporters are basically the cellular doorways, right?
Exactly. They're specialized proteins embedded in the cell membrane that pull specific nutrients inside. If a microbe has a massive translational efficiency for a specific import protein, scientists can see clearly which specific substrate that microbe is desperately trying to pull inside.
So if we see a microbe is bleeding ATP to build, say, ribose doorways, we know without a doubt it's actively starving for ribose.
Yes, and because we know the functional guilds, we also know that if we drop ribose into the environment, that specific microbe will thrive, and its direct competitors are going to suffer.
The researchers tested this precise mechanism right, feeding their 16-member syncom specific substrates. When they added ribose, Two microbes, Burkholderia and painted bacillus, spiked significantly, and we know exactly why. The TE data showed they had incredibly high priority for ribose importers named RBSA, RBSB, and RBSC.
They were the primary targets, yeah.
But the critical part is, because those two primary targets spiked, their specific guild competitors, Variovirax and Rhizobium, were structurally starved out and decreased.
And they observed the exact same mechanism with different nutrients. When they added a diamine called putrescine, rhodococcus, and painobacillus spiked. Consequently, their guild competitors, Rhizobium and Bradyrhizobium, decreased. Feeding one specific microbe its preferred substrate automatically suppresses its rival.
You know, there's a great mnemonic for the Mindic algorithm here. Measure TE, infer competitors, name the food or niche, and direct the intervention. Measure, infer, name, direct, mind.
That's a brilliant way to remember it.
So feeding the right substrate boosts the target and starves the rival. Think about your own body. What is your personal high TE import protein? What's the one specific food that always gets your energy up? But wait, a pristine lab flask is one thing. Does this actually survive in the wild? The real world is a jungle of unknown variables.
That is the ultimate test, and it's where this paper gets truly groundbreaking. They moved from ex vivo models into highly complex, undefined environments, starting with a soil microcosm. They took wild switchgrass soil, thousands of interacting species, and added our probiotic friend Catenofaga along with its preferred prebiotic glutathione.
A highly targeted prebiotic-probiotic combo.
Precisely. The result was a 13-fold increase in Catenofaga right there in the wild soil. Simultaneously, they saw a crash in its predicted guild competitor, Mucilaginibacter. But even more impressively, MindDive predicted the behavior of the native soil microbes too.
Wait, how did the native microbes react?
When they added complex carbohydrates, native Clostridium bacteria spiked. And just as predicted, as Clostridium surged to consume the carbs, its specific native competitors, Azospirillum, Bosacea, and Streptomyces, were outcompeted and driven down.
Here's where it gets really interesting. They moved directly to the human gut. They extracted bacteria from an 8-month-old infant stool sample. The primary goal was to see if they could predictably boost a highly beneficial bacterium called Bifidobacterium longum.
Right, a classic probiotic strain that we constantly try and often fail to reliably establish in patients.
So they ran the metabolic on the stool sample. The TE profiling revealed that B. longum's preferred dietary niches were the sugars xylose and fructose. When they introduced xylose to the complex stool community, B. longum experienced a massive 160-fold spike. Fructose caused a respectable 3-fold spike.
And they mapped vitamin niches as well. By adding cobalamin, which is vitamin B12, they caused another beneficial microbe called Vascularctobacterium to spike 19-fold.
A 19-fold spike just from a vitamin is incredible. But they also looked at adult human gut samples, and MIND revealed something that completely shifts how we view current probiotic treatments. Two of the most famous beneficial gut microbes, Faecalibacterium prausnitzii and B. longum, are actually in the exact same functional guild.
Meaning they are direct competitors.
Yes. They're fighting over the same plate of food, which perfectly explains why so many clinical trials end in confusion. Researchers feed a patient a complex prebiotic hoping to boost both, and one increases while the other plummets. They had no idea they were pitting them against each other.
It highlights why guessing based on correlation is a dead end. If you don't know the functional mechanics, your interventions will always yield chaotic results.
Okay, time for another 5-minute recap. Bullet 1, transporter TE reveals exact dietary niches. Bullet 2, targeted combos work in wild, complex soil. Bullet 3, MindD successfully and predictably boosts targeted human gut bacteria.
Perfect.
The leap from simple lab tests to complex human microbiomes is huge. If we can predictably boost good bacteria in an infant's gut, what other human health conditions could we theoretically program away?
Well, to truly prove this can cure human disease, it has to work inside a living, breathing body. That leads us to their in vivo testing. They used a mouse model targeting a poorly characterized mouse-specific gut microbe called Faecalibacillum rodentium.
Which we know helps with glucose homeostasis, right?
Right, but historically, we haven't known exactly what to feed it to make it flourish.
So they run the protocol. Metabolic on the mouse gut shows that this specific microbe absolutely loves a sugar called D-lactose. And the delivery method they used is brilliant. Adult mice naturally lack the enzyme lactase in their intestines.
Yes, meaning if an adult mouse drinks lactose, its own digestive system cannot break it down or absorb it.
It's the ultimate stealth delivery system. The lactose bypasses the host's digestion entirely and goes straight down into the lower gut, perfectly intact.
Exactly. So they put a 2% D-lactose solution in the mice's drinking water for 14 days. The result was a selective, massive enrichment of effordentium right inside the living animal. And crucially, it did this without throwing the rest of the gut into dysbiosis. They selectively tuned one single dial.
It's an incredible proof of concept. But I do have one lingering question. What happens if two completely different microbes have an equally high TE for the exact same food? Like we saw earlier that both Burkholderia and Rhizobium prioritize building doorways for ribose. If you drop a single molecule of ribose between them, who actually wins? How does the framework predict the tiebreaker?
That is a phenomenal question. They accounted for it by integrating a computational grading tool called GEM's genome scale metabolic models. Think of a GEM as a massive mathematical simulation of a cell's entire internal metabolism.
How does the simulation calculate a winner, though?
By analyzing something called flux balance. Imagine two competing car factories. They both have the exact same priority for acquiring raw steel and pull it in at the same rate. But flux balance calculates how fast those raw materials can move through the internal assembly lines. The GMM simulation looks at the internal enzymes of both microbes and calculates their theoretical growth rates.
Oh, wow.
Yeah, and in the case of Burkholderia and Rhizobium, the GM accurately calculated that Burkholderia simply possesses a naturally faster internal assembly line for processing ribose.
Ah, so they both want it and successfully pull it inside, but Burkholderia literally metabolizes it faster, turns it into energy faster, and therefore outcompetes Rhizobium purely through sheer metabolic speed.
Exactly. The models break competitive ties based on who can physically process it faster. Survival of the metabolically fittest.
So MIND works in living mammals, and computational models can break competitive ties. When two organisms want the exact same resource, it comes down to sheer metabolic processing speed. How does that change your view of survival of the fittest? It's not a mystery, it's just an incredibly complex equation of energy.
If we connect this to the bigger picture, it means the era of treating the microbiome like a black box is officially over. We no longer have to guess with prebiotics and probiotics. We don't have to take a pill and just cross our fingers. We possess the tools to rationally engineer microbiomes based on functional mechanistic truth.
So what does this all mean? We've gone from tracking tiny fragments of active mRNA inside a bacterial ribosome to identifying hidden competitive guilds to ultimately manipulating a living mammal's biology just by adding a specific sugar to its water.
It is a fundamental shift from passive observation to active engineering.
We just learned how to target a single microbe by feeding it its favorite sugar. If scientists can map the TE of every microbe in your personal gut, could we eventually design a single personalized meal that completely rewires your immune system or metabolism overnight? Imagine food as literal targeted code.
It is a profound and increasingly realistic future to consider.
Thank you so much for taking this deep dive with us. Tomorrow, when you sit down for breakfast, take a second to look at your plate and wonder which microscopic guild are you actively feeding and who is losing the race.